On behalf of BioIT World's Global Web Symposia Series and our sponsor, Flywheel, I'd like to welcome you to Unlocking Precision Medicine, Streamlining Data Management for Multi-Site Traumatic Brain Injury Research. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I would like to introduce our presenters for today. First is Geoffrey T. Manley, MD, PhD, Professor and Vice Chairman of Neurological Surgery, University of California, San Francisco, and also Chief of Neurosurgery at Zuckerberg San Francisco General Hospital. Our second speaker is Sina Aslan, PhD, Scientific Advisor at Flywheel. Welcome, Dr. Manley. The presenter ball is yours.
Thank you very much. I appreciate the opportunity to speak to you today. What I'd like to do is to give you an example of a large multi-center study of precision medicine, which we started about 10 years ago with a variety of pilot studies culminating in our current large network, and to really kind of describe why a solution like Flywheel has been very important to the progress of our network and our continued success. So the problem that we study is traumatic brain injury, and many of you have become aware of traumatic brain injury, and some only in the last 10 years, even though this is one of the oldest injuries known to man. And this is due large in part to understanding more about when folks were coming back from these conflicts in the Middle East, that we had a number of these soldiers who were no longer dying, but because of the life-saving value of things like Kevlar body armor, et cetera, they were sustaining what we call traumatic brain injury. In addition, I think we became aware that things like concussions aren't always benign, and actually much of what we see even in a level 1 trauma center is concussions. They're just not from sport. They're from people riding bicycles, people falling down steps, et cetera. And so there's a number of people, and we don't actually really even know the exact number of folks that sustain a concussion every year. It's been estimated that there's about 4.8 million people that seek medical care in the United States each year for traumatic brain injury. And despite the fact that we've tried all kinds of ways to mitigate these things with seat belts and airbags and so on and so forth, human beings are really, really resourceful when it comes to finding other ways to hurt themselves. Our most recent trauma issues seem to surround these scooters that you can get on the street simply with a cell phone, and people don't wear helmets when they ride them, and we get somebody almost every day that comes in from one of these types of accidents. So trauma is happening. Trauma is never going to go away, and we need to learn how to deal with it. The other issue is that we've done such a good job in medicine taking care of patients with heart disease and with cancer that one of the fastest growing segments of our population are, as you see here on the left, elderly folks who are falling down steps. Everyone is much more active today with the incredible advances that we've had in heart medicine and as I said earlier, treatment of certain forms of cancer and joint replacements and things like that. And so we have many people in their 80s and 90s that are much more active than they would have been, say, for example, when I was a kid. And so with this rise in what we call the silver tsunami, by 2030, there's going to be 7.72 million older Americans that are out there. We've seen this in the statistics from the CDC, and so what you can see here on the left is what things look like in 2002, and then roll forward to 2013, and new statistics are now coming out. It takes a while to process this data for the latter part of the 2010s, if you will, and this trend continues. We had looked at this in the state of California and found that, in fact, there were about a 40% increase in traumatic brain injury over the past 10 years in California. And much of this is being driven by the geriatric population and, fortunately, increased awareness about traumatic brain injury. So it's not just a U.S. problem, at least 69 million people each year worldwide. So this is a huge issue, which many of us believe that while we have the opportunity to rapidly advance the field, given the tools that were developed in other disease groups, we're still quite a ways behind cancer and heart disease, and our patients really need for us to advance this as quickly as we possibly can. So when we look at traumatic brain injury and the way that we describe this, we have classification systems that use terms like mild, severe, and mild TBI, and severe TBI, and concussion. And one can see that those aren't very precise at all compared to the way we would talk about a stage 2 breast cancer tumor that's BRCA1 positive and receptor status, et cetera. So we've still got a ways to go. And even how we look at patients' outcome, historically, we've looked at things like death or full recovery, and again, not very precise. And, we've now begun to understand, particularly as we've paid more attention to what we presumed were less severe injuries that we called mild TBI, but in fact, they're really not. And so we realized that there's things like, depression and cognitive challenges and post-traumatic stress, et cetera. So there's a lot of things that we're just now learning about traumatic brain injury. And what we wanted to do, starting about 10 years ago, was to really take what we call a precision medicine approach to TBI. So what do I mean by that? Rather than using terms like mild, moderate, severe, we wanted to be more precise. So not just simply working in this realm of symptoms, but also really trying to harness the value that's in imaging. And this is really where our relationship with Flywheel currently stands, and why it's so important, because imaging really drives much of the decision-making in traumatic brain injury. And so to be able to archive these images and to store them and to be able to process them in certain ways, not for clinical care, but for the clinical data that we acquire, and then to be able to process that to gain more insight is very, very important, and it has been a challenging problem for us on the data management side. In addition, the clinical data, even though we have electronic medical records, most of the information that we need for traumatic brain injury patients actually doesn't exist there, and so we've been trying to work on how we would kind of glean that information from that. We do a lot of work in the realm of the proteome, so blood-based tests for traumatic brain injury. We were fortunate earlier this year to participate with Abbott Diagnostics, and they received the first clearance for a handheld point-of-care blood-based biomarker test. So that's moving along, and also looking at the genome. So what you can see here is that there's a whole lot of disparate data that we're trying to capture with these studies. So what is the goal of TRACK? Well, we're trying to look longitudinally. It's an observation study in a precision medicine sort of way, trying to come up with better diagnostic or classification models for this, to really try to develop better tools for outcome assessment. And at the time, while we've been writing a lot of papers and it's no longer underrepresented in the field, at the time that we wrote this grant, really trying to define what the health and economic impacts of what we called mild TBI, and we've certainly been making great strides there. And then to create this information commons that could promote collaboration and acceleration of TBI research. And my colleague from Flywheel will be talking a little bit more about, where is this imaging management relate to this idea of this information commons. So TRACK is currently at 19 sites around the country. We're getting ready to add 5 more sites. It's a very active network and a number of diverse sites. As you can see here, we have a large team. I'm just speaking to you today representing our group, but we really represent the best and the brightest in traumatic brain injury research across the spectrum, from the pre-hospital arena all the way out to long-term care and rehabilitation. So there are members of this team that really touch every different part of the patient trajectory and care experience. And many of us many of us were competitors on prior grants, and now we find that our collaboration is really allowing us to achieve things that not one of us individually or one university could do. So it also speaks to the need to have data management tools that aren't just in-house, not just something that's in your lab or in your university, but we need a tool that allows us to be able to ingest data from all of these sites and have input from all of these team members. And what I can tell you, from struggling to try to find the right solution here, it's been a real challenge over the years. So we have lots of data. We have 3,000 subjects with traumatic brain injury. We have orthopedic trauma controls. We have friend controls. We have over 3,000 data fields on each subject. This is a tremendous amount of information. We do 21 outcome measures at repeated time intervals of 2 weeks, 3 months, 6 months, 12 months. And each one of these outcome measures can have, up to 40 or 50 questions in the questionnaire. So you can start to see the depth of how we characterize these patients. But it's the brain, and the brain's complicated, and you can't just simply look at something like, are they able to go back to work? Because you really want to know why. Is it because it's a mental health issue? Is it because they're cognitively challenged? What's actually, stopping them from returning to work? So these disability measures don't really tell the whole story. And then here, as it really relates to what's going on with our relationship with Flywheel, we have a tremendous amount of standardized adult MRI scans. And I want to note here that with these scans were obtained using research protocols. So the de-identification of these images is a little bit more straightforward. And, we have these at repeated intervals at 2 weeks and at 6 months. Whereas the CT scans that are done as part of standardized care, these images actually are rife with PHI and things of this nature, just because of the way that we work with them. And I don't have time to go into this in detail, but I could follow up offline of what are the challenges of de-identifying a CT scan. So what this means is, having tools that help us with the de-identification, and in particular having storage or be able to create a repository that meets all the regulatory requirements that we'll touch on a little bit later in this is very, very, very important, because I understand that people want to share data and they want to be able to work with other folks, but we also have a responsibility to our patients to protect their personal information. And this remains, I've been doing this now for 20 years, this remains a very, very challenging aspect, and in a place where I think Flywheel has developed some interesting tools to do this. Lots of blood-based biomarker specimens, brain donation, et cetera. So lots and lots of things. So if you put this in context, this is a slide from a colleague of mine at the NIH. And what we have here is showing, sort of the evolution of big data within traumatic brain injury. And so you can look back to some of these earlier studies from, 2015, and you can start to look at, how much data is being generated, And then if you look at the top right over here, you can see this is our TRACK-TBI study, which we still are going through validation steps at Pittsburgh to be able to allow other investigators to work with this data set. But this is probably the largest, most granular data set. Certainly our care colleagues data set, they have more subjects, but we go a little bit deeper into each of the subjects in terms of the granularity. So it's a very, very large data set. And while we really are happy that we've collected all this data, I will tell you what we feel like on a daily basis. And that's really, just struggling with the complexity and the granularity. We believe that these kinds of data sets are going to drive machine learning and AI and a lot of things that we're all very, very excited about. But the capture and the curation and the management of this is really probably the biggest challenge that we did not anticipate when we set out on this effort 10 years ago. And I showed this next slide here, and it's very true. I mean, we feel like almost every day we're drowning in the data. I think that we were very naive when we, set out on this and not fully understanding how much it was going to cost to curate, how much it was going to cost to clean. And, we still struggle with this every day. And it seems like every time we try a new project, we find a new problem. And as one of my colleagues, Mary Vassar, says, big data has lots of little problems. And that's why you really need robust solutions, particularly for multi-center work, to be able to, again, acquire this data, to curate and manage it. So with this, I'm going to turn this back to our colleagues here at Flywheel, and then they'll tell you a little bit about how this application's been built to help our particular data needs. I'll turn it over to you now. Thanks.
Thank you, Dr. Manley. Hello, everyone, and welcome to our webinar. My name again is Sina Aslan. I'm Scientific Advisor with Flywheel. Today, I plan to give an overview of TRACK-TBI workflow in Flywheel. As Dr. Manley pointed out, TRACK-TBI Consortium's goal is to identify biomarkers of traumatic brain injury in order to accurately diagnose and provide clinical treatment, and ultimately achieve precision medicine. So the right set of tools are needed for this huge undertaking. Having a capable, scalable bioinformatics platform to provide data and compute management is key. So what is Flywheel? How did it come about? About 10 years ago at Stanford University, our founders noticed that researchers were performing cutting-edge research with data technology, wasting a lot of time and energy. Researchers were capturing the data on CDs and DVDs, jump drives, and then uploading it to local servers. Every lab was siloed. There's no easy way to share data or algorithms. Researchers are spending a lot of time on curation of the data itself. They also noticed lack of continuity in their research labs. As members of their labs moved on with their careers, the extent of the analysis was not captured. Provenance was lost. So the whole Flywheel idea was born. So what is Flywheel? Flywheel is a secure, HIPAA-compliant biomedical research platform that offers both data and compute management to support clinical research and AI. At a high level, Flywheel has 4 major categories of capture, curate, compute, and collaborate. We call it the 4 Cs. In the capture feature, a connector, which is deployed locally, would be able to, for instance, ingest the data, DICOM or non-DICOM, from scanner and PACS or a file system for a fully automated capture. Unlike research packs, Flywheel can accept any data type, even the ones unknown to the system, very much like a file system. You can also integrate with EMR and ingest metadata from electronic data capture systems, such as RedCap or Question. In the curation stage, the platform would perform classification, quality control, and you can tag files and images so it can be searched for later on. So this whole capture and curate portion is a data management portion of the Flywheel. As we all know, the real scientific work really begins after the data is captured and curated. So the compute management is an important part of the Flywheel application. For instance, having the ability to interact with the system live through Python and MATLAB or R software for developing algorithms. Finalizing and uploading algorithm, we call them gears in Flywheel, and enforcing version control for scientific reproducibility is also key. The execution of these gears is also important. You can run hundreds of jobs simultaneously, route these jobs to a high-performance cluster, or even a specific cloud provider with a specific virtual machine is important. All of this is capturing provenance. Who did it? What was the configuration? How it was done? And the last feature, of course, is a collaboration. After all, scientists need to collaborate, such as TRACK-TBI, with one another. And they find it hard to share data and algorithms while respecting IRB regulations. Flywheel provides an easy mechanism for users to share their data and algorithms in a secure, collaborative environment. All of these features have made Flywheel an appealing platform in the imaging, research, ML, and precision care apps. By the way, Flywheel can be deployed in the cloud, on-prem, or as a hybrid model. TRACK-TBI is fully deployed in the cloud, thus it gives the consortium the necessary scalability for storage and compute needs. So how's Flywheel helping TRACK-TBI with data aggregation and curation? Let's review their workflow. So as you can see, I learned that there's now 19 sites contributing data to Flywheel. Each Flywheel instance has been set up to receive and have only access to their own projects. Each site can upload using their own custom de-identification, as Dr. Manley mentioned. That is an important thing, especially for CT. In order to make sure the data is uploaded by each site is complete, the project admin would set up a session template. Also, a set of gear rules. Gear rules are a set of automated algorithms that get triggered when a specific criteria is met. I'll be covering this in more detail shortly. Once the data is complete and has passed QC, then the data is moved or copied to a main project and is ready for analysis. In parallel, each site is entering their own cohort's clinical data into Question. As part of the next step in our implementation, we will be working with the TRACK-TBI team to ingest these metadata in question into Flywheel, so there is a single platform to look up both clinical measures as well as imaging data. Now that all the imaging data and clinical data are in Flywheel, each site can perform search in their own projects and find both imaging and clinical data. Additionally, project administrators can give appropriate access to their collaborators to search, create a collection, which these are soft copies of the data for sub-cohort analysis, or simply download the data for further analysis. In the next set of slides, I'll be covering this step in more detail. So there are several challenges, as Dr. Manley mentioned, for having a shared infrastructure. The burden of implementation usually is on a single entity, thus the host entity enforces IT requirements, usually lacks customizable permission, and the access logs are not very granular. There's nothing beyond entry and exit. On the other hand, Flywheel is a commercially managed solution that offers secure login with each user's institution. No need to create a separate username or password. Customizable permissions beyond admin read, write, or read is configurable in Flywheel. We capture all user actions in access control. Now, the first question is, how do I access the platform? Well, users will be able to access the platform simply via a browser, nothing to install locally. Once the user logs into the URL, he or she is presented with our university login option, which directs the user to a list of over 4,000 universities. Once a university is chosen, in this case UCSF, the page will be directed to UCSF's login page, where the user would need to comply with his or her own university's IT requirement, such as two-factor authentication. Once validated, Flywheel will ensure that that user exists in the system, then displays a dashboard with that particular project that he or she should have access to, in this case, a site 1. TRACK-TBI has several different ways to upload their data. They can simply drag and drop their studies to a web uploader that we have, ideal for ongoing studies as you acquire the data. We have a command line interface, CLI, which is a small executable file for uploading the data in bulk, in orders of gigs or even terabytes. Soon, we'll be deploying our connector so that images at UCSF can directly be routed from this scanner to Flywheel automatically. Now, all these three methods will respect this custom de-identification that we talked about. In the data curation stage, the goal is to classify the data, convert the DICOMs to nifty format, which is common in the neuroimaging field, and perform some sort of a QC. Some of the challenges that multi-center studies such as TRACK-TBI faced before was automatically checking for incomplete data, automate or standardize image pre-processing, variation in scan protocol names, or lack of provenance. Luckily, Flywheel has a solution for all the above challenges. TRACK-TBI project admin would define a template to make sure the data is complete. A set of gear rules, again, these are algorithms that would get triggered based on a set of criteria, can be defined. They can also standardize and customize data classification. So you no longer are bounded to remember all these different protocol names. All the above activities are fully captured with provenance to ensure that derivative data is fully reproducible. Once the data has passed the session template, fully QC'd and classified and pre-processed, now it's ready for further analysis. Typically, data scientists have a specific hypothesis that requires them to search for a subset of data in order to analyze or train their models. This is where the data scientists at TRACK-TBI will be able to search for their data or metadata. One of the challenges that data scientists face is the inability to find data or metadata, or at least it's very hard and difficult for them to do so. For instance, DICOM tags need to be read and correlated to Excel files to obtain clinical data. On the other hand, Flywheel offers a powerful search functionality that could look up data such as images as well as clinical data that's been ported over from Question. There are two types of search functionality, basic and advanced. In the basic search, a user is presented with a basic predefined facets, such as modality, classification, file types, to name a few. And then user can choose any of these and narrow down their results based on the projects, of course, they have access to. Now, in the advanced search, on the other hand, users can construct complex queries similar to SQL commands, which would include more operators like or, contain, or define ranges if they need it. So the ability now to securely collaborate on data algorithm is also very critical, right? Flywheel platform has been developed to be HIPAA GDPR compliant to meet IRB regulations. We also offer 21 CFR for any FDA submission. As part of the challenges of the data and algorithm sharing, data typically is locked into clinical systems. It's hard to get out. They need to be de-identified. Logging and querying changes are not captured. And it is just difficult to share, right? Data in ML typically is very large. And then algorithms have a lot of dependencies, ranging from the operating system itself to the program that you're going to be running. So it's very environmental dependent. In Flywheel, we recognize that as far as the data, ingesting the data goes, it not only should be easy to ingest the data, but exporting the data should be simpler. If every project can be downloaded in Flywheel via the UI with one click. We also understand that it is important to capture all the changes, right? Provide a detailed access log for each action to not only meet regulatory compliance, but also support scientific transparency and reproducibility. Flywheel allows also sharing the data by giving custom permission at the project level for collections. These are soft copies of the data. And then at very last, we also recognize that sharing algorithms is important. So containerizing algorithms or gears allow the users to share these algorithms within the platform or across platform. So all of these actions support full provenance and governance of the data. Some of the future directions that TRACK-TBI could take and utilize Flywheel for is, for instance, in clinical trials. We can facilitate secure file transfer and verification, custom de-identification from those sites, data collection, processing, or sharing, all under 21 CFR Part 11 compliance for FDA submissions. We already have external viewer connectivity, such as Osiris or 3D Slicer, but we could expand to other viewers if necessary. We also support blind reader study workflows that are very much important in clinical trials. To summarize, we showed that Flywheel was able to accommodate TRACK-TBI not only with their research needs, but also to provide an efficient and collaborative environment to expedite their research. With that, I'd like to, on behalf of Flywheel team, I'd like to thank TRACK-TBI, particularly Dr. Geoff Manley and Pratik Mukherjee, for their partnership and opportunity to expedite their research. We look forward to having similar partnership across different disciplines and to assist you with any of your research needs or endeavors. Thank you.
Super. So I think they're going to turn the ball back to me to just go over a couple of some of the advances that we have made with some of our infrastructure here. And I want to just go over a couple of preliminary findings. So as you've seen with the presentation from my Flywheel colleague, we've really taken advantage of many of the features of the Flywheel system to both ingest and to manage our data. And I just want to just roll back just a little bit. One of the main things that we've done in TRACK-TBI is really building upon the successes of ADNI and some other imaging studies in the past is that we've been able to standardize a lot of the imaging that we've done. So we use the ADNI structural phantom, we use the FBurn functional phantom, and then we actually at scale deployed the ice water phantom that was originally developed by NIST, and we have publications on this which you can go back to. I think it's also important here, we want to leave enough questions for time for questions and so on, but I just want to be clear, I should have mentioned this in the beginning. I have no ownership in Flywheel. I own no stock in Flywheel. I have no conflicts of interest with them. They're simply a service provider for us for this aspect of our study. With that disclosed, I'll share a little bit of the work that we've done here. And this was published in August of 2020, looking at the evolution of white matter microstructural changes after mild traumatic brain injury. And this is looking at comparison of longitudinal diffusion tensor imaging with this new methodology that many of you may be familiar with called NADI. And one can go to the paper to look for all of the data here, but I just wanted to show you just some examples here where we're looking at comparisons of folks with mild TBI, which we define as GCS of 13 to 15 with orthopedic trauma controls. And I think if I were to look at much of the literature that's out there, too often we're simply studying the disease and we don't have enough controls. So in TRACK, we have both orthopedic trauma controls and we have friend controls as well, which give us a little bit of a normative population, which are age matched to these subjects that were enrolled in this study. And so what we found is that these early decreases in fractional anisotropy and the increases in mean diffusivity do correspond to the elevated free water fraction, which is termed FISO on the NADI imaging modality. And importantly, this elevated FISO in subjects was associated with symptoms inventory on the Rivermead post-concussive questionnaire. That's the RPQ. And this is probably has one of the biggest effect sizes when we look at an outcome measure in this population of GCS 13 to 15. And importantly, these white matter changes were associated with declining neurite density on the NADI imaging, suggesting that there was axonal degeneration from DAI or diffuse axonal injury, for which NADI appears in our hands at least to be more sensitive than DTI. And this was done in a pilot sample of patients, and we're now validating this finding at scale. I'd also like to say that, there's been a lot of focus on MRI imaging, and I think that's where Flywheel really got their start, was working with MRI imaging. But the workhorse for traumatic brain injury is actually the CT scan. Most patients with TBI only get CT scans. Right now, MRI scanning is in the realm of research, although we would advocate that patients with traumatic brain injury need MRI imaging just like patients with stroke and other neurological diseases. But we're really not quite there. But interestingly, while we've been spending a lot of time looking at MRI imaging over the last decade, CT imagers have been quietly evolving. And so most of us have multi-slice scanners. We have much thinner slice thicknesses. We look at this in both axial and coronal and sagittal planes, and we find much more pathology, I think, than we looked at in the past. That center panel there is from one of the old early generation CT scans that some of us grew up seeing, in the early 80s. So I think that there's been a lot of evolution in CT. And so we went back and examined whether or not these imaging findings that we were seeing on CT scans, and we see them quite frequently in these patients with GCS 13 to 15, which we have historically called mild. What is the clinical relevance of this? Because there's been a lot of debate about it. So this is a paper that was published in July of this year, looking at the pathological CT features that are associated with adverse outcomes in what we call, quote unquote, mild traumatic brain injury. And so what this is what's called an upset plot. So this is kind of like a Venn diagram, but in a different way. And so if we just look at the top figure here, you can see the largest bar there is for isolated subarachnoid hemorrhage, which those of us who do this clinically, that was not a surprise, is that isolated subarachnoid hemorrhage is the most common thing that you see with positive imaging with people with a GCS of 13 to 15. But then there's this co-occurrence of both subarachnoid, subdural, and contusion that's quite common. And so what we did was looking at this, it was clear that this wasn't random, that there were patterns to this. And could we use a data-driven approach, extracting these features using the TBI common data elements to say, which of these things actually go together? What are the clusters of CT imaging phenotypes that one can see in this population? And so what you see here on the left is a dendrogram doing just hierarchical clustering. We've done this with multiple correspondence analysis, and it identified, three dominant groups here. One is, epidural hematoma, which we know mechanistically is due to an injury to a blood vessel on the dura. It's not actually within the brain where that injury occurs. And those seem to be an independent entity, whereas things like subarachnoid hemorrhage, subdural, and contusion are more consistently found together. And not surprisingly, the contusion subarachnoid hemorrhage is very closely found together, but that's associated with subdural hematomas. And then, on the far right of the dendrogram, you can see intraventricular hemorrhage and PH, or petechial hemorrhages. On an MRI scan, we would call those a microhemorrhage. Those that can be seen on CT scans are indicative of more microhemorrhages that one would see on an MRI. But these sorts of injuries are due to rotational injury. And so there was some sort of validation there if we went back and looked at the biomechanics of traumatic brain injury as it was described by Ayub Omaya and Tom Gennarelli now 40 years ago. And when they were doing primate work, these are really the predominant injury phenotypes that they found. So that we felt like this replicated some of the prior preclinical work, certainly fit with much of our clinical experience with this, and really started to think about some of these are more sort of linear acceleration, some of these are more rotational, and then what are the consequences of this. So, excuse me, I'm going to just go back one slide here. Just to sort of finish this out, I don't want to go through our statistical approach. We use generalized estimating equations. We looked at this at repeated measures over time. And the upshot of this is that with the exception of epidural hematomas, when you find subdural subarachnoid hemorrhage contusion on a scan, it's associated with more unfavorable outcome in this population than those that have a normal head CT. And in particular, IVH and PH, or petechial hemorrhages, is associated with even more unfavorable outcome than those lesions that we just showed above. So I think the take-home point here is that when we see these, even in patients that have a normal exam or a GCS of 15, we can't just simply dismiss these people as mild TBI, because in a very large study, as I said, done at multiple sites, we demonstrated that this is associated with unfavorable outcome. More importantly, these findings were validated in an external data set that was done with our sister study called TRACK-TBI in Europe. And so we did this in about 2,000 patients. Center had about 2,400 patients. So this is a very large sample size, suggesting that this is in fact a true and real result and merits clinical adoption of better management of these patients in the future. So, just to summarize my experience of how Flywheel has helped us, I found this picture online. Here's some guy carrying some old hard drives and stuff after some disaster, which is kind of how we feel when we're deep in the curation of our data sets. But I mentioned this because much of our data transfer prior to working with Flywheel occurred through actual physical transport of hard drives. We were working with another entity where we were trying to transfer things online, and there were these huge jobs that would run for days, and then it would fail, or we would board, or we'd have a problem with this or that. And so finally, because these things needed to be transported back to the East Coast, we would just start, when I would travel back to the East Coast, I would simply carry a bag full of hard drives to move our images back and forth. And if we wanted to get images to a collaborating colleague at another university, we just load up a hard drive and send it off. And I think that that's really, things of the past, and very inefficient, very time consuming. And I think that, with Flywheel, we have a much better platform in which to do this, where, we don't have to consume time and effort copying stuff, send it to people, they download it. There's really, as as we just saw, you can create spaces where people can access certain images and not access others. So this has been a real advance for us. And I think the other thing that was mentioned in the previous talk about containerization of these algorithms, it's not just standard acquisition of the imaging. That's very important, and you need phantoms and you need all that. But it's also trying to achieve some standardization with our analytic pipelines. And when you containerize something, it actually facilitates that process. So not only does it make it more efficient, because not everybody writes code and is a data scientist, an imaging scientist to be able to do this, but when you have things that are containerized, then you can replicate results more easily. So rather than trying to deal with some clunky algorithm, if it's containerized, you can take your data set and you can rerun it, and you can then validate that. And as we all know, there's a crisis of not being able to validate these results. And in particular in imaging, we have all kinds of studies that have differing results, and this is in 75 patients there and 80 patients there. And so the ability to not only standardize acquisition, but also using these containers to standardize some of our analytic pipelines, I think will add to better research in the future. So I really thank you for your time and effort to join this today, and to hear a little bit about TRACK-TBI and how Flywheel has helped us. And I think we'll be happy to take any questions that you might have. Thank you.
Thank you very much, Dr. Manley and Dr. Aslan. We do have some questions that have come in. Our first is for you, Dr. Manley. In your experience, what are the biggest challenges with data sharing for large-scale research networks?
I'm sorry, can you run that by me one more time? I apologize.
Certainly. In your experience, what are the biggest challenges with data sharing for large-scale research networks?
Well, so I think I highlighted a couple of these things. One is just simply getting the images to collaborating colleagues. So that's been a challenge. I think the other issue is as I said, some of these images, particularly some of the clinical images, for us, the CT scans have been very difficult to de-identify, and so that's been a challenge as well. I think that looking at things like material transfer agreements and legal barriers, I think, or at least certain legal issues that one has to get through with this is also difficult. One of the things that we're starting to look at, and where I think that these containerized gears that Flywheel has that may offer us a new way to do this, is that if you look at the paper that we did at JAMA Neurology, where we externally validated these results in TBI, they did not send us their CT scans. Their CT scans had data that was already extracted using TBI common data elements. And what we did was we did federated analyses. And so it may not be necessary to always send your images out. It might be possible, say, for example, that if, you're in a challenging material transfer agreement environment, say, for example, of where things are today between the European Union and the United States, someone might actually build a gear for you, and you could just go ahead and run that gear or that container on the data that you have in Flywheel. And if you can replicate the same results on the data set, then you've replicated it. So I think there's a number of ways to do this. There's still many, many challenges to how one collaborates and works with these things. But I think as these tools get developed, we'll have better solutions to facilitate that in the future.
Thank you so much. A question for you, Dr. Aslan. My institution is looking into a research PACS. Is Flywheel a research PACS? If not, what makes Flywheel different from one?
Flywheel is definitely much more than a research PACS. As I kind of alluded in my during my talk, research PACS are very limited. They have, they can only hold DICOM data. They cannot have, for instance, non-DICOM data. And for precision medicine, as Dr. Manley mentioned, you have your clinical outcomes, you have your genomics, and all these other types like physiological data that needs to be integrated with your data. So even the data management portion, how Flywheel handles that, doing cohort based designation is definitely much more than what research packs can do. Plus, you have a lot more capability. We call it like basically it's a super research pack. We have the compute side of the whole thing that you can execute algorithms with version control on these large amount of data that you have collected. So Flywheel is basically a perfect solution to replace research packs and to provide not only the data management portion of it, but also provide compute management and a collaborative environment, while all of that under a HIPAA compliant regulation.
Thank you very much. And this question is for Dr. Manley. Daniel asks, how did using Flywheel help overcome challenges you faced early on or with previous projects? And what do you see as the biggest benefits to using it for multi-site research?
Well, again, I mean, for those of you who are on the webinar today who've done this in the past, there's been some pretty clunky systems out there to do this. And so I think that, there's been models where there will be an imaging repository that's done at another institution, for example. And so we try to get all those things to another institution. And then if you're trying to access that, through that other institution, then the issue is that, it can be difficult because a lot of times these folks do these projects with multiple other studies because it is very expensive to run something like this. And, it's not always easy to get access to the individuals that you need to help to manage the data because you're not directly managing it. So I think for us, one of the benefits here is that we have an instance, we have all the data that's loaded into that. Our investigators can access this data and we can manage this data more directly than some of our previous models for doing this. So the other thing that I would say that got brought up as well is that, as you're doing this, even for whether it's for a clinical trial or it's actually for high quality research, I think that all of these images, and this is my personal opinion and others may agree or disagree, but I think, what we try to do for all these studies is we have multiple readers to look at this, and the systems that are built in to do things like, automatic adjudication and having multiple readers, to do that without software is actually challenging. And in the past, we tried to build a software system to do that, and it works okay, but it's not great. And so, we've had multiple challenges, and this product grew out of an academic group too. So they were suffering from some of the same issues that we were, which is why I think that at least for us, and I suspect for other academic groups, they'll find that this meets a lot of the needs that people have and also solves some of the problems that I think we all were experiencing. And again, no tool is perfect. But I am not directly overseeing the management of this tool. I'm the contact PI for our group. Dr. Pratik Mukherjee is the lead of our imaging core. And, we talk on a regular basis and this is working for us. I certainly, prior to adopting this tool, it seemed to me that from someone trying to help to oversee and steward this large project with, 75 different people on it, that I was fielding a lot more emails about, issues with imaging than I've been dealing with since we adopted the Flywheel solution. So I guess, if that's another metric, which I think it is, it seems to be working for us. And for me, as the sort of overall manager of the project, I'm having to deal with less issues because of this.
Perfect. Thank you so much. And Sina, Brian asks some questions regarding what non-imaging data types can Flywheel store. For example, could it be used for digital pathology data or even genomics?
Yes, so Flywheel can store any types of data, even the ones that the system doesn't know about. Now, those file types that Flywheel does recognize, let's just say, NIFTs, DICOMs, PNGs, or maybe even EEGs, brain vision. We can parse out the information from these files, attach them as metadata so it becomes searchable, and we typically have a viewer for all of these data types. Yes, we do support digital pathology as well. We are happy to give a demo if you'd like, but the system is also extensible to where you would be able to upload your own algorithm. We have an exchange of over 100 gears, mostly focused on, I would say, neuroimaging. So you can use those, or you can containerize your own algorithms and push it to the system. So if you'd like to have a viewer, if you don't like the way we are showing you a pie chart or showing you a Nifty file, or even you have an image viewer for maybe genomics, you can extend the platform to do that. And we are happy to provide a demo on Digital Path for sure.
Excellent. Thank you. And going back to Dr. Manley, what do you envision as some future uses of the imaging repository and biorepository?
Well, I think that, we're still in the process of just barely scraping the surface of this data set, and we have, a variety of collaborators where we're working on things. We have a very robust collaboration with some of our colleagues at Lawrence Livermore National Labs and Argonne and Berkeley Labs. And so I can see us, continuing to delve into the value of imaging and, Flywheel helping us to support that sort of activity. For the biospecimen repositories, of course, that's more challenging because there's only so much biofluid that one can get from a patient, and it's not a never-ending resource. Whereas the imaging, once it's actually curated and put into a robust data management platform, one can use that data forever. So we continue to think about what's the next generation of solutions where, not only would you have something like Flywheel, but you could bring in your genetic information, you could bring in your blood-based biomarkers, all the clinical phenotyping, and really what does the future of a precision medicine data cube really look like? And, certainly that solution, unless somebody that's on the webinar knows about one that I don't know, is not out there. I think we're working on base solutions like things like Flywheel, which are really, from our perspective, as I said, less emails, we're getting more work done, and that's really accelerating our research. So I think the future is bright, as we see a lot of advances in technology just in general, and I'm looking forward to continuing to use this solution and waiting for other solutions that might bring some of these. We still have several databases that we're using. There's not one database to sort of rule them all. And that I think is due to the fact that we've got a bunch of complex and granular data that we really probably don't have quite the same architecture that's needed for the future. But we're very happy where we are. We're definitely working better than we have in the past. So I think as we look to the future, this will help us to fuel our collaborations and get more work done.
Thank you very much. We have a couple more clinical questions for you, Dr. Manley.
Okay.
The first one, you had mentioned in your previous answer about biomarkers. Have you found any blood-based or imaging-based biomarkers for traumatic brain injury?
Yes, we have. So I would direct you to a couple of papers. There is a paper by David Okonkwo, which was published, I think, last year, looking at the association of GFAP and CT imaging. We've had some prior work with GFAP and UCHL1. Those are the two biomarkers that have actually been cleared by the FDA, and the objective biomarker that they used to clear that was CT imaging findings. We also have a paper that was published in Lancet Neurology. The first author is UAYUE, and this shows that even when CT images are read as normal, and then one finds that they have abnormal findings on a head CT, which in our situation in a level 1 trauma center, about 25% of the people with normal CT imaging will be found to have abnormalities that are indicative of traumatic brain injury, particularly microhemorrhages and some contusions. in about 25% of the population. And interestingly, we see elevations in GFAP that are associated with those MRI findings. And we have ongoing work now with neurofilament light and some other biomarkers. And I think in total we may have looked at about 100 different biomarkers, but currently, the biomarkers that we're really focused on are some of these FDA cleared biomarkers, neurofilament light, and some others as well, tau, p-tau, et cetera. So there's an exciting area there, and certainly the alignment between the blood-based biomarkers and the imaging biomarkers is very exciting. Thanks.
Excellent, thank you. And looks like we have time for just one, maybe two more questions. This one is for Dr. Aslan. How does Flywheel help researchers with machine learning? What tools and functionality are available to help with this?
Yes, absolutely. This is actually a question we get all the time. So as we have heard, is that more than 80% of the ML developers spend a lot of time on data curation. So one thing that we have is this whole standardizing classification. For instance, in TRACK-TBI, you have one site naming T1 a protocol, you have T1W and then T1 weighted. Now, all of these are the same thing. So we just classify this in a standard way. We call it, it's an MR protocol, structural, T1, maybe it's an NP-RAGE protocol. Now, that gives the user, ML user, and the ability to search on these, right? You go and search, I'm interested in all the NP-RAGE or T1 weighted images. Now, that search would be narrowed down to that particular subject, session, or a specific file. I mean, if you're wanting to grab that nifty file for that T1 weighted image, you could do so. So you create a collection. A collection is a soft copy of the data. And meanwhile, maybe you want to bring in another clinical outcome or some sort of a demographic information. You want to grab age, maybe with some GSW that Dr. Manley mentioned, scores, right? So now you can create a machine learning training data set in form of a JSON file, where this would be a snapshot of what is available in the system across all the projects that you have access to. Now you can run your model and run your pattern or whatever features you need to extract from by using this JSON file, because every file or object in Flywheel has a unique global identifier. Now, given that these ML data sets are somewhat large, maybe you need to grab the data, but you're just trying to develop your algorithms. You're not quite ready to containerize it and push it to the system. You can use our Python SDK to interact with the system rather than copying over gigs or terabytes of data to your local computer or server. You can interact with those files, maybe one at a time, store it in the matrix, and then run your model through it. Once it is finalized, now this is the time you containerize your algorithm and push it to the system so that other users, maybe nobody, and not everyone is a programmer, as Dr. Manley mentioned, so they can run it with point and click or just set up a gear rule for it so that every file, for instance, that comes through, it can run through it automatically. And by the way, we do version control. So very much enforce software versioning so that the next time you or maybe your colleague want to upload the next algorithm, the same name to the platform, the system will force you to increment the version. So now you have full provenance attached to all of these jobs that get executed. So now you know what were the input, what were the outputs, what were the configurations, along with the log file as if you're sitting in front of the terminal. I hope that helps.
Yes, thank you so much. I see we have run over our time. We do have a couple more questions. I will forward those along to our presenters and you'll receive those answers via email. I'd like to thank our presenters today, Dr. Geoff Manley and Dr. Sina Aslan. I'd like to thank the folks at Flywheel for sponsoring today's web symposium. Most of all, I'd like to thank those of you who came and spent this time with us. We're very grateful that you came. It's a very challenging time and we're being pulled many directions. So we're very grateful you came and spent this time with us. So on behalf of BioIT World's Global Web Symposia series, thank you all so very much and have a great rest of your day. Bye-bye.